speaker this morning, Dana Ballard. And let me start by sharing. And here we go, share. Oops, I didn't want to do that.
Don't worry, we won't look.
Let's see. Can you see my screen?
Yeah, but we're seeing,
Yeah.
let's share. Let's start the presentation. Okay, good.
Okay, so this is actually before that slide that Toby showed, which was the very first Telluride meeting in 1994. Well, that was preceded by an NSF workshop in Washington, D.C. that took place, as you can see, in 1993, August 27th. And NSF asked Christoph Koch, Rodney Douglas, and me to organize a group. This was in the heyday of neural networks, and the idea was, can we somehow get the biologists together with the engineers? And that was really, as you can see, these are some of the participants, and Richard Anderson, for example, was at MIT at the time. Apostolos Georgopoulos, who was a colleague of mine when I was at Johns Hopkins, was at that point in Minnesota. Steve Lisberger, ocular motor system at UCSF, Tony Movshon, visual system. So that, there was a lot of systems people here, but look, the ones that I've outlined in red were the ones that actually showed up at the first 1994 meeting. And so that with the planning that we did at the workshop led directly to the Telluride meeting. But how we actually got to Telluride was itself a real long story that I don't have time to tell, except to say that it was very fortuitous that we found this box canyon, which is incredibly beautiful, and very, very produced 28 years now, if you include Zoom meetings, of great workshops. So here is that inaugural meeting. As you can see at the time, the building was a high school, but in fact it had been turned into an elementary school later. And if you look through here and identify some of the people, you realize that a lot of them are still coming to Telluride. So here's Timur, for example. Here's Shishi, and here's Rodney, and here is Dana in the back. You can see him with the mustache. To his right is Toby, and to his left is Christoph Koch. So this really is a historic photo. And I want to tell you a little bit about Dana. I've known him going back to the 80s. In fact, we wrote a Nature paper together, parallel models of visual processing with Jeffrey Hinton. And in it, what we tried to do was to come up with a new framework for thinking about vision that went beyond the tradition at the time, which is to come up with very low dimensional geometric models to try to fit it like templates, and that kind of had its heyday in that era. And our goal was to say that, look, let's think in terms of massively parallel processing, in terms of many units interacting with each other, and the algorithm is going to be now the connections between the units. And Dana had already gone down that path, very, I think, as Toby was saying, thinking of it not in terms of image analysis, which is what Dana at the time called dead-eye vision, where you have a dead image, but rather active vision, in which the animal has to interact with the world in order to be able to seek out information and to visual motor tasks. How do you accomplish a task? You have to move your eye around. You have to move your head back and forth. That gives you information about depth, right? Motion parallax. And that was really a turning point in vision, is that, vision is an active sense. It's not a passive identify the object problem, although obviously that's an important problem too, but it's not the one that is important for interacting with the world directly. And let's see, I think yeah, I want to point out that Dana wrote one of the most popular textbooks in computer vision very early on, 1982, with Chris Brown. And this textbook actually had a huge impact on the field because it was very well written and it really covered a lot of topics. And I want to point out, somebody already did, that if you want to buy a copy now, you're going to have to fork out $900. But even more telling, look at the date here. January 1st, 1656, Dana goes way back to the 17th century in terms of his thinking, right? It was clear that he was way ahead of his time. Now you can go on the Amazon website and see this. I'm not making this up, right? This is it. This is what you see there. And actually, this is not a really good picture of the book. This is a more realistic picture of the book. And I want to point out this again, 4.5 stars at Amazon, but look at the spine here. It's clear that whoever used this book had basically, changed the color of the spine just from all of the sweat on the poem. There you go. Okay, so Dana is he was at the time when I met him at Rochester. And in fact, he was one of the cabal in the early days of neural networks that called themselves the Connectionists. And we used to meet for workshops in the summer at Rochester, Pittsburgh, and elsewhere. And it really, a lot of my early, thinking was formed in this small group in terms of the interactions we had with each other. And it was really very exciting. And I have to say, he's been a lifelong friend in terms of being a compatriot from the neural network era. And he's gone on, he's moved to UT Austin, where he's continued. I'm going to point out just one example of a paper that has had a large impact on today's research. So he had a really prescient paper with Raj Rao, who was a graduate student of his, on predictive coding in the visual system. How what the visual system is in the business of doing is predicting top down what's in the image, and then looking at how well with the mismatches between them in order to be able to come up with better predictions and be able to understand in a generative way what's out there. Okay, with that, I'm going to hand it over to Dana. And by the way, one other thing you should know about him: he's an avid sailor. I think he still has a sailboat, and he's has spent a lot of his time on the ocean, on the eastern seaboard. Okay, Dana, take it away.
All right, here we are. Let's see. We need to get to my share screen, I guess. Here we go. And we get to the talk, and that is here. And we start. I would say thanks for Jerry. Yeah, I worked in vision for a long time, but I thought I moved to Texas in 06. I thought, well, if you ever want to do something new, now's the time. And I thought motor control, I didn't know much about it, but I thought, how hard could it be? And I also thought people would be very excited to have a new person in the field.
Dana, you just reminded me of an event that occurred at the first Telluride meeting along those lines in terms of motor system. So Dana he didn't bring just a bunch of people. He actually brought a go-kart that had been instrumented and with an eye tracker and so forth. And this cart, this huge cart, a crate, shows up with 2 holes in it from a forklift that had gone right through the crate. Do you remember, Dana?
Yeah, I do that. I remember the hole. And
Dana was thinking, oh my God, this is, what's left inside, this. And so we opened it up, and miraculously, the 2 forks from the forklift had gone right through empty spots in between the pipes and the go-kart, and nothing was touched. It was a miraculous, and that just gives you a sense of what it was like to be in Telluride at that time. But motor systems coupled with vision is definitely what Dana was already thinking about back then.
All right. Well, thanks, Terry. Well, thanks for the introduction too. We are friends for lifelong fellows, and we've had a lot of great times. But I won't tell you itemize them here because we do have my talk to get to, which is motor control. Is very. This is proven to be extremely interesting for me. And we built a number of things. And one is a system that basically can integrate mocap data and stuck on a model. And if you have a model of a human, a 50-degree of joints with human, you can recover the torques of the model, and that's very exciting. And so that's going to be the topic of today. And then a lot of the students who worked on it have graduated and they have jobs. But we have Lee. Here we go. We have Lee Deer. She just got her PhD, but she did some beautiful work on this, and it's going to be featured today. So here we go. What I've done is thrown away all the equations. So we're just going to have fun. And here we go. And if you wanted to look at human motion, the thing that just hits you over the head is it's so complex. And we'll start with all the muscles. So over 600 muscles and 300 degrees of freedom. So it's mathematically, it's very, very, very challenging. But I love Lena Ting. She's in Emory, and she has a beautiful way of triaging the issue. And one is, if you want to start on human motor control, the biggest thing you get first right out of the box is your skeleton. So it's underappreciated, the fact that all of the joints and this construction of it is in place to be a prior to the kinds of movements that you would make. It's just the movements that you want to make. Not only that, the next level up is going to be muscle co-contraction. And this is really a monster ability, because you can make yourself into different tools. When you make movements, you can be very different dynamical system depending on what you have to do. Try to do that with your robot and see what happens. And finally, the interesting thing is, when we have all this in place, what should we do with it? We want to make movements, and what's the strategy for getting the neural system to come up with something that'll do the job? So, okay, one is another cautionary warning for us is that motor turns out to be very, very complex, and almost every system you have in the brain has a little projection on some issue in movement. But here, I've tried to take the textbook picture and make it simple. And what do we have? The pink and the green. And the pink is the forebrain proper. And what's going on here is for us to think about. It's open loop. What does that mean? That means that the somehow, and now this is not solved, somehow all the movements you want to make are in there, and that if you want to make one, you just pick one and send it down to the rest of the system. But now green is closed loop. So you have a raft of closed systems that trying to help. It'll turn out that the open loop is not perfect. It's good, it's very close, but it's not perfect. And you need to you need your close loop systems. And if you want to test yourself, stand on one leg and close your eyes and see how long you can balance on one leg. You can read off your age by the seconds you took. Okay, let's remember this: open loop, closed loop. Okay, and now you're getting the score. Whoop. I want to go back here. You're getting the score talk. Spoiler alert, because this is the whole talk in one slide. And so if you remember these things, you got it. So each one, of course, is a very, 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 very point. And we'll start that when you make movements, that's been a lot of people study human movements, but most of them are reaching and walking. If you had those papers, you would be able to paper in every wall in your like pretty big size apartment for those. But we want arbitrary movements where you just do whatever you want, and whole body movements. You're not sitting in a chair like I am now looking at a screen. You're walking, dancing around and lifting something. All kinds of things are happening. And so we want to, and here you got in the first one is the bottom line. Amazingly, subjects would choose different subjects, ask the main the same movement, choose identical trajectories. How could that be? Okay, we'll go there in a moment. The second thing is, given that the kinematics are the multi-system human body movement, if you have that, then the modern, many groups are doing this. If you have the kinematics and you have a body model, then you can get joint torques and just by straight up direct integration. So that's like a breakthrough. And we use it too, and other do groups, but that's a big one. Okay, but now you know what's coming, because if subjects are using identical kinematic trajectories, and kinematic trajectory is all you need to recover the torques, then you can tell people what a movement costs. And it turns out that, and so let's back up one sentence and ask ourselves, if all the subjects are using the same kinematic movements, why are they doing that? The obvious answer to that one is because they are somehow cheap. So that somehow your brain not only makes the movements, but of course you could change the things with weird constraints. But if your everyday choices are on the table, what you're choosing is cheapest ones. So how do you figure that out? So this is my Ligia figured out a way to do that, and we'll take a look at it. And now, wow, we have the package. And so why are we giving a Telluride-like talk? It's because if we have all the minimum movements, why don't we just store them in M1? And a lot of people say, you can't do that because there's too many. But we think that's not true. So there we go. So let's move on here. Okay. So here we go. Well, this is kinematics, of course. We know how we get that. Somebody wears a mocap suit, and then we have a model of there on the left. And so we can basically get a description of that. And what are we going to do with it? Here's an exotic thing we're gonna do it. We're going into a virtual environment, a dojo with, and we're gonna wear a helmet with eye tracking in there so we can see what's happening in 3D. And what's the task? We have these big tasks, they're very big tasks, and you can stand wherever you like, and here's this white dot, and you have to put your finger on the white dot, and it'll race along the curve, and you have the red dot, and you sort of have to trace it. So if you're too bad at this, you can't be a subject. But most undergrads are really good at this, and so there they go. Here we go. Whoop, 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 whoop. You'll notice the curve is really big, so you can't stand in one place. You have to follow it around. Oh, good job. Oof, maybe one more. Are we gonna have a C one? Yeah, maybe this is an easier curve, but there we go. Whoop. Do. Oh, very nice. Very nice. Okay, that was beautiful. Now, here's our curves. We're going to have 9 curves, and there are different sizes. And then we're going to trace all of them. All the subjects, 18 subjects, all are going to. 18 times 9 is the data, and you're going to have their kinematics for all these paths. And so what do we want to do? Well, the first thing is you put on your suit, and you when you have the data, you're a graph. So the data is in the graph, and so you have a posture is all the links on the figure. And what you want to do is the crucial thing is if you have different postures, you want to know how far they are apart. So if you have the graph from one posture and the other one, you'd like to know how far apart are they. And Ligia came up with a measure that works really well for doing that. So now this is the hardest graph. So if you get this one, the rest is going to be easy. But what are you looking at here? This is the tracing time. And when you have the posture, let's take frame 300. And so all the subjects have the postures. And so when we take the distances between them and the mean, they're going to have a distribution. So here's the mean posture. You take all the postures and take an average. And then the rest of them perform a distribution. And that's here. Okay, that's great. And we can see that all the postures at frame 300 are pretty closely clustered. But now let's take 520, and that's for the subjects that traced that was their mean. But now what we're going to do, we're going to take that mean, and we're going to take these postures and make a distribution from using these means, but these postures. And so these postures here are far away from these ones. So these ones are very about here, but if we put it there, well, it's not a good fit. But you can see astonishingly that this works for every place in the curve. So if I say take the data from 300 and try to match it against all the other places, nothing matches. But it turns out that you can do this for any point. I could take any frame and try to match it to the rest of the things. What you see is that it's very, 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 very significant here is the matching the postures at the place they belong is much cheaper than matching everywhere. And so what this is telling us, when knocking us over the head, is the postures tracing this curve of people. These people are not all, the data is not normalized for height and other parameters. It's really rough, but they're very, very, very different. So, but that's not all. Is let's take the data, and if we have the means, we could take the variances. So what we can do is, for all the points in the curves that are traced, and here are the 9. These boxes are the different trays. All these are tree curves being traced here, and this is pi. And we don't have all the subjects. We have every color is a different subject, so you can count how many we have. But what you see here, okay, what are we going to do? We're going to, as we trace at every 10th frame, we're going to compute the variance of the distribution, and now we're going to sort them, the variance from smallest to biggest. And now we're going to plot it for all the subjects. And what you can see, this is very exciting for me, is that what you see is not only the means colored, but the variances are correlated too for all the curves. And what we have a hypothesis on the table that we're testing, but the idea is that the variance is related to the co-contraction. So if you're co-contracting, the variance goes down. If you don't care, it goes up. But it's very exciting. So not only are the curves identical, but the variances are correlated too. So let's see. Wow. And so, so what are we going to do this? We would like to get to cost. And so the way we're going to handle this is we are going to make a human dynamic model. And Joseph Cooper, a former grad, did this, and Lee Jira cleaned it up for him and made a thing of beauty. But it basically has 50 degrees of freedom, and it's built on the ODE. And Joseph had to do a lot of surgeries on ODE to make it friendly. But so that's our system, and it has 15 degree of system, and it's especially built for people, humans. But you can get into amazing cat fight anytime you want by comparing our system to the other OpenSim, Muju, Joko, Hamburg-Eppendorf. They're all, these are competitor systems, but we would have an argument of why this is good. You'll see why, some of the reasons why we do. So let's see. And one thing Joseph had, he had to clean up the joints for the human to make it human-like. And that's one thing he did. And then here is his system, and this is like a summary, all the joints, and basically the degree of freedom for the joints. Okay, this is a summary of the human system here. It's ready to go. But now let's take it out for a drive. So Joseph did a very nice thing. It's very, very creative. So the people, to get this data, they wear a suit. The suit has markers on the seat. So what he did in the dynamic ODE, he gave the markers on the suit infinite mass, or let's say a very large number, and he built it so there's every suit marker has a companion on the human model, and they correspond. So every marker in the suit has a marker on the model. And so what happens is, and they're connected by stiff springs. And so when something is with infinite mass moves and you're connected to something with a spring, you're going to come along for sure. And so, but what to come along, the dynamic model has to make torques in the joints, and it turns out that the torques that it makes are the same torque you would have to make if you just started without markers of any kind and just wanted to make the movement. So let's let this go. Here we go. So first of all, we have to make the points correspond. And yeah, okay. And now let's integrate these things. What are those torques? Okay, well, if we do enough steps, we got them. So now we don't know we don't need the suit anymore. Let's take the suit off and play the torques. Okay, well, oops. The suit's moving and the model's moving. What went wrong? It turns out, just for the humans, that the open loop torques are not good enough. And so you need some sort of closed loop in here. So we're putting into a faux vestibular system here. And what Joseph is turning is turning up the gain and giving it more value. And if he gets it right, you can walk off into the sunset. This is very, I don't know, he's a very creative guy. And Joseph think he was a great dancer, but here he is with his PhD video. And basically what's happened is that's Joseph, the model is doing this. So you got the mocap and you plugged it into the model, you added the vestibular control, you're done. We can move on. Okay, and wow, motor control, what a vicious field, they said. There's no evidence that it's computing anything. So, but Joseph had the model stand on force plates and lift a leg and another leg. And you can see that the stance things are very, very, very close, or close enough for government work. So it works. It gives you very nice representations of the torques. I think we can move on. Now, the other thing you have to know, and this is Ligia played with this and tuned it to perfection, is that if you might think that, well, I'm building, if I want to control this, what I should do is use all the degrees of freedom in the model. It turns out that that's a very bad thing to do numerically. But what you can do is you can, and this is well known in the motor control literature, that if you you can pick a subset of the control joints, and then the dynamical system is all stuck on it with connections, and so it'll do the right thing. So if you want to make a movement, you can have to demand every degree of freedom to do the right thing. Just pick a subset, and you'll be fine. And this is a beautiful curve for some of the joints, showing that if the dotted line is with the reduced control, and the other one is the law data. So here we go. It works perfectly. Okay, so now what are we going to do? And this is we talked about that. Well, of course, we have curves, and so we could let our guy, he could trace the curves, because he has a model, so he knows how to do. Okay, wow, good for you. All right, so here we go. One graph using the cost. So here's something you might expect. So you don't have to wake up yet, but you can see the length of the path. It goes from the shortest to the longest, but you can see the cost of tracing that there is also monotonically related to the length of the path. So that's kind of a sanity chest. That's pretty, we saw it. But wait a second, we could do things, and this is Ligia's idea, this really good one. And there's two perturbations. So well, let's see. So we're going to perturbate the paths and the model and see what happens. So let's do the model first. So you have the poor little guy, you just saw him, the model in there. He has to trace the thing, but he has to keep his elbow up a small amount during the trace. So he's tracing as before, but he now has to keep his elbow up. So that's one thing we could do. Or what about the path? We could take the paths and then just move to the left, move to the right. So the model person has to trace it, but you have to keep your feet on the ground where they were. But let's see what happens with that. So this is the model, and this is the thing of the graph you're looking at is a thing of beauty. Is that for all 9 paths, it takes you if you're going to try to hold your elbow up while you're doing this, it's going to cost you. And so what this graph is showing you, the extra cost of the perturbation when you subtract off the constrained perturbation here. Okay, so I can't look at this without my heart beating faster. It's very beautiful. And so this is a lot of data, right? This is all the subjects are tracing different paths. So a lot of data. And this is nice too. This is, now we're going to move the thing to the left, right to the right, and then green is right and blue is left. And you can see that, well, there's a bad boy down here. But again, with all miles and miles of data and a little more variance, everything's above the line. Sure, that all the time you trace with the almost, when all the time you traced it, the shift curve, it's going to cost you. So,
But can I ask a question, Dana?
of course.
Why would just the translation of the path cost more energy? You're just moving it over to the left or right a little bit. Why would that cost more energy to trace it?
Oh, well, I mean, when you trace it the first time, you sat in, put your feet down in a place that was good for your stance, right? So you had to, if you had to move a little in, under it or whatever you want in for your little piece, wherever it would be good to stand, that's where you go. But you're not allowed to do that in this perturbation. You use the same foot plan.
Okay, clear, thanks.
Okay.
So Dana, I have this, Andreas, a follow-up to Toby's question. So this is a perturbation on the body too. So you're really perturbing the body stance to really determine this. You don't allow the body to move.
The body can body can move however it wants, with the constraint that you're going to use your same foot plans.
Oh yeah, that's what I meant. Yes, yeah.
But what happens above the ankle is
right. That's what I meant. Yeah, sorry. Yeah.
Okay. So if you were on Ligia's PhD committee, you would say, well, this is yet a perturbation. What about this doesn't show that it's you use the minimum trajectory in what you do. But I caution this guy's in computer science, but that in fact, once you leave computer science, everybody accepts this perturbation methodology.
No, that's beautiful. It's nice. Yes.
Okay, well, let's move on. And this is a beautiful thing. To open any medical textbook, but it's this is the motor strip and the some sensory system. And everybody knows this map is there. I was on a NIH site visit once, and these guys had a magnet stimulator. He had it a Tesla, one Tesla. He had it over his fingers, and he blasted himself, and he could play his fingers just by doing that. And I thought, wow, these guys are desperately for money. But what I love about this is it's wrong because of Michael Graziano, because he had a beautiful thing that he did. And Georgopoulos did things like that much earlier, and other people too. But the idea is that if you can use higher stimulus in motor one, you'll get whole body movements. So you're not just tweaking a finger or a tongue. You can, and this is Graziano's thing. He classified them into about 5 classifiers of eating, defending, and et cetera, et cetera. And but the bottom line for us is that if you just stimulate in a location in M1, you can get a whole body movement, and that's going to be very important for us. So, and here's a paper I love just to read and look at this. But here's the Stanford group, and what they did is there a monkey's in a chair. The monkey has to hold his fixation, and then a target comes on, and he has to hold his fixation still. And when he dims the target, the fixation, then he can reach, or she can reach to the target. And this, I love this. There's 3 trajectories here, and between the dots is 20 milliseconds. And what happens is, okay, when the target appears, yes, oh, what did they do? They had about a Utah array with 100 neurons, and they did PCA for the 2 axes. And here are the axes of that cabal of neurons. And you can see that when the target comes on, you're not done. So you have to waddle around in PCA spot, and then the dim comes, and you're still not moving. So in green, you're now get something with the target, and then finally you get to the light purple, and you can move. And so I challenge everyone to figure out what's going on here. But my answer would be is that you're looking up the movement. You're not making the movement. You have to just look it up in there. It's in there. So let's see. So let's. I'm just gonna assume I have enough time. Okay, but here's it. Let's just have this. So basically, we have these curves that we in M1, and we know that from Graziano that somehow we have to have a little piece of a trajectory. So we have to. How are those trajectories coded? Well, let's let this is Casey Green, and he's using OpenSim. It's one of the simulators, and the colors are on the muscles, on some of the legs, they just put in for the demo. And so he's an undergrad, he's having fun. Okay, but now let's look at those muscles, the same movement plotted where the muscles are plotted around their resting length. And oh, wow, this is just I've gone to the opera. This is like music. This is fabulous, totally fabulous. Here's the muscle of the movement you would think. The movement's very arbitrary. But let me move us on. So this is all, everybody in the audience understand this is only too well. I'm going to use basis functions that is going to be these Gabors, and here's some parameters that you could use, and okay, we'll do a little sparse coating. And so here's one of the muscles, and for 2 seconds of it, and I picked out one of my basis functions, and it's pretty good. Ooh, better. And whoops, I'm doing that. But let's go back. So this is what have we seen is that the coding is fabulous because if you go to the conventional robot crowd, they would control things in real time. And well, how much frequent? What do you need? What's the bandwidth? And 40 kilohertz is a number that floats around. Now, here's for the Telluride crowd. See if the brain has enough bandwidth to control 600 muscles at 40 kilohertz. You can't get even close. So the movement has to be coded. But what did we learn? We learned that once you have the kinematics, you can just send it to the spinal cord, and the spinal cord will integrate it for you. So you don't have to code every little torque at any little time. You just have to code the kinematics and tell what the spinal cord what it is, and you can do movements. And so you know the punchline is coming now, is that since we have these coefficients for the kinematics, we actually could code them in M1, all the movements we want. But I would say only the minimum energy movements are candidates, and grumping in the audience. But they would still say, look, come on, do you still have too many movements all the movements you would make? But you have to take into account another constraint, which is that you don't have to do this hours ahead. Movements happened in real time in let's say 2 or 3 little intervals. And so you could do all the intervals. And if you have a hairy movement, then you can make them a necklace by stringing the ones you want together. QED. So now I have my slide, which is the last one, and thank you. And I also thank my friends and also the people who kept Telluride going. But it was just a fabulous place. It's just where people who are very talented just run around and have fun together. And it was, and maybe, and also Terry, of course, he never gives himself enough thanks, but he was really he was the mover and shaker for this. So thanks, Terry.
Okay. Well, thank you, Dana, for a very stimulating lecture. I'm going to start out with the first question, but I want others to jump in.
Then I have a quick question, Terry. Dana, are you going to come to Telluride next year?
I hope so. I would love to. I mean, I feel Telluride deprived. And I will definitely come if I can.
Well,
and is Dana going to come? Is Dana going to come? That's the
Dana.
Oh, Dana, are you coming to Telluride next year?
We assume you're going to come, Terry. We don't question that.
We just assume that.
Yeah. Okay. No, Dana, you have an official invitation to Telluride next year.
Well, actually, the bottom line is, you might have said, got from this talk that I'm very, very excited about this, but it turns out that there's something I'm even more excited about in face coding for the neurons in the cortex. And I have a neurocomputation with my brilliant student, really brilliant, seriously brilliant, is Rohan Jean. And that is a sort of a opening stance for this. But I'm very, very, very excited about that, because I think the spike coating in the forebrain has to be totally reworked, and it's totally open, and we've taken some steps in there, but that's the thing I'm really, 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 really excited about.
Well, we are looking forward to that talk next
And
summer.
a work group.
Oh,
Yep.
it may actually, you could give that talk. Yeah. So Andreas, why don't you schedule it? This, you know.
It's going to be tough this time. Yeah, we only have a few days left. But Dana, listen, I remember you shared me your nice neural computation paper. I admit I didn't do anything about it, but I would like to share it on the Slack for comment from the crowd. Is that okay, or you're eager for debate about it, right?
Oh, sure. I have to say, I had a hell of a time getting it published, and Rohan, my student, he said, you know, we have to republish this. So he rewrote the paper and sent it to neural computation, and they loved it. So there you go.
Okay, so we'll see what other people have to say about it.
Okay. So here's a question which connects what you've done, which is very elegant, and you know, the whole body motion is clearly many degrees of freedom. It's beyond what robotics people do, I think. I mean, you know, we're still not up to that yet. Robots look very stiff. They only have a couple of joints going at a time. But one of the things that comes out of those experiments with single or double joints is that you can get a trajectory with minimum jerk. And how does that, which is acceleration, right? Square acceleration. How does that relate to minimum energy?
Well, I'm sure that there would be some sort of relationship, but I think I don't, frankly, I don't know what the sales pitch for a minimum jerk is. I think at the time it was just when people are thinking of all these local things that the computer could compute, it was one of the options. But I think I hate to give such an uneducated opinion, but I think it's dead on arrival. But I'll think about it, Terry. On your probe.
No. It was all done with curve fitting. In other words, it's a Gaussian, and then how do you get a Gaussian from minimizing what functional and you know.
Okay.
You know, it's all curve fitting.
But Terry Sinofsky, I heard from some robotics people that notion is not so well used at least in robotics. Minimum jerk principles. I don't know. Again, I'm speaking from ignorance.
Oh, no, you may be right. Well, maybe they discovered energy too. Who knows?
Okay. Anybody? Anything else from the audience? Not right now, but ask your question. Let's see if there are any questions.
I have another question for Dana. Okay. So there a lot of discussion about eye movements early on, but you didn't say anything about eye movements, during the task where the red ball has to, in other words, you were tracking eye movements too, right? Wasn't that part of the?
Yeah. I haven't thought very deeply on it, which is a career thing, but it would be that it's important. And what I think is there are a lot, if you're going to index these things in M1, then what are you going to use? And so a lot of the movements can use fixation as the origin or something of the movement. So I'm sure it has a role in terms of the movement thing. Oh, and by the way, I'm at another movie that I didn't show, but because I didn't want to go too long. But my colleague Mary Hayhoe is doing totally fabulous work these days and she has subjects walking over outdoor rough terrain. And he shows that they have a model that shows how the fixation picks out places where you're going to step. And the model is, it can work. So you can use walking outside, you can use gaze to pick the place you're going to use for your footfall.
So that turns out to be true for cats as well as humans. I saw a movie just a couple of days ago of somebody put a cat cam, you know, on a cat was walking through a dense forest, and what the eye tracker was able to fixate where the paws were going, but every once in a while it would jump to a distant location, you know, where it was heading on the trail, and then back. So it's clear that it was multiplexing.
Yeah. So well, it makes sense, right? That with the stereo vision, you know, it's going to have to. Yeah, I think vision really is right. It does the planning. It's the distal sense. So it's going to do the planning of what's
here comes John Doyle with some comment. I hope. Anyway, we're out of time, right? It's a really great introduction from Terry. Great talk, Dana. I'm sure it's going to get lots of views. I hope you are happy with that on YouTube. Sure. And now we have to pass it over to the TAC group, and Emre will take over the hosting. Are you there, Emre?
I'm here.
Big thanks to both of you guys. Fantastic.
Thanks for
keep in touch, Dana.
being audience. That's great. That's great.
All right. So our next talk is going to be from the tactile group, and Kara, I think looks like you are there to present the speaker to us.
Yep. Hi, Emre. Hi, everybody. So today we have the pleasure to host a talk by Stefano Panzeri, who is a computational neuroscientist who works at the interface between theory and experiment. He investigates how circuits of neurons in the brain encode sensory information and generate behavior. He graduated in theoretical physics at Turin University and then did a PhD in computational neuroscience at SISTA in Trieste. And he took faculty and adjunct or visiting faculty positions at several institutions in the UK, Italy, and the USA, at Harvard, and in Germany. He currently works as full professor and director of the Excellence Department of Neuroinformation Processing at Hamburg. So Stefano, the stage is yours, and thanks a lot for coming.
Thank you, Chiara, for the introduction and for the invitation. Can you hear me? Can you see my screen?
Yes, well, we don't see it, the presentation in full screen.
Yeah, it's not, okay,
PowerPoint.
Now you should see it in full screen presentation. 1 second, okay. Do you see my presentation?
Perfect.
Yes.
So thank you very much, and good morning or good afternoon or good evening to everyone. I don't know where you are, if you're all on site or not. And Chiara asked me to contribute to this tactile group. So I will present some of the work that we did over the years to study, I mean, the sense of touch, information coding in the sense of touch, and in particular, the role of spike timing. And this is a long-standing interest of mine, but just to give you some background for the people that don't know me, I'm a computational neuroscientist. I don't do any experiments, but I work in close collaboration with experimentalists. I'm particularly interested in sensory coding. And I started my work about 20 years ago on the sense of touch. But over the last few years, I moved to study other senses, especially olfaction, vision and hearing. And at the end of the talk, I will give a very brief overview of maybe 1 minute, 2 minutes about how the finding that I present here for the somatosensory system in my work translate to other sensory modalities, if that is of interest to you guys. Okay, so I mean, the problem we study is the one of neural coding, that is how neurons encode and transmit sensory information. Particularly, we try also with other people to try to address in the somatosensory system what is the language or code used by neurons to transmit information to one another. So operatively, this is essentially amount to understand which response variable encode information, or in the case of spike timing that we're going to talk about today, it was the temporal precision needed to read out neural information. And I mean, it's quite clear, I mean, to everyone that the neural code is a sequence of spikes, because all these spikes propagate long distances. And from the work like that of Zach Main and Teresa Inoski, we know very well that neurons can generate even in vivo and in vitro and vivo, I mean, very precise sequence of action potentials. And the question is whether this very precise millisecond level sequence of action potential actually carry information which is used by the brain. And I mean, the reason why I feel it's interesting to study these questions, also for people not maybe that work in neuroscience, but also the people who work the frontier in neuroscience, maybe also in the community of neuromorphic engineering, is that characterizing this stuff, so how neurons encode information, what are the precision, time scales at which they operate, what is time scale carry information, it's useful to understand brain function, but also to understand how to model neurons in network, for example, rate model versus spiking neural models for any given application that you want to do, and also to understand how to connect engineering device with the brain. So I think this type of work is potentially useful for those applications. And the general, I mean, so the question that we try to ask is whether the timing of spikes carry information about tactile stimuli. And I think this kind of plot that I show here is useful to illustrate the typical approach of neurophysiologists. And here you see a picture of a rat snout. And here you see some histology showing the groups of neurons that encode information about the whisker, those are called barrels, because in 3D they have kind of barrel shape. And whenever you stimulate, for example, if you place an electrode in barrel D2, which is one of those barrels correspond to the whisker D2, you get like a strong and early response from this neuron when you stimulate this whisker. So if you stimulate other whiskers, for example, two nearby whiskers in the same row, you get maybe lesser spike numbers, but also longer latencies. And what people do, they look at various responses, for example, the number of spikes, I mean, the latency, the structure of spike intervals, and they try to build a kind of a Rosetta Stone between the sensory stimuli that you present and the neural responses that you obtain. And I will discuss in the later part of the work the limitations of this approach. But I think it's very useful to try to construct a kind of dictionary or Rosetta Stone that connects the stuff that you don't know, which is how neurons encode information, the language used by the neurons, and the external world, which is something that you control as an experimentalist. And the spike timing hypothesis, the one that we try to, to report here what we investigated in the somatosensory system. So this hypothesis states that you may have modulation of firing rate by spike count that carry information, but also the exact timing of spikes at which the spikes are emitted is important. So it carries information about sensory stimuli, tactile stimuli in this case, it is not conveyed by the spike count alone. So not only spike timing should be able to carry information, but also information that you cannot possibly find in the spike counts or spike rates alone. So in this case, for example, that I showed you before, you might think that definitely spike counts carry information because they are modulated by the stimulus, but also you see that latency is modulated by the stimulus, but you might say that maybe latency does not add any information to that of spike rate because latency is correlated with spike rates. It's shorter for stimuli that elicit a stronger response. So to prove this hypothesis and kind of demonstrate that spike timing is useful to encode information, you would need to demonstrate that not only the spike times are reliably modulated by the stimulus, but they carry information that you cannot find elsewhere in neural response in longer time scales. So, the thing that we try to prove over the years to understand these spike times are crucial for neural information processing. As I said, we did this work in several modalities, but we report here only about touch and motor sensation. I think you need to prove 3 points. So the first point is that they carry information that is not found elsewhere in a slower description of the neural response, for example, in spike rates average over, say, tens of milliseconds. Then also they should carry information in a format that is usable or decodable by another downstream neural system. I made the example of the latency before. Some people believe, rightly or wrongly, but I think it's a good point, that a latency is measured with respect to the stimulus presentation, and maybe user experimenter may know about it, but another neural circuit might not have this information, so might not know about it. And they should also have a measurable impact on behavior. So you should be able to prove either by causal manipulation or statistical analysis that whenever there is a variation of information in spike timing, you have also corresponding variation in the behavioral report about what is perceived. And so the first point to address is whether these guys carry precise spike timing and the second spike timing carry information that is not carried by spike counts. And I give you here a very brief overview. We work a lot on this with Matthew Diamond, who is a neurophysiologist. So most of the data here are recorded from Matthew Diamond's lab, and some of those at the end by Tommaso Fellini, a colleague at IIT where I was before. 1 second, sorry. Okay. And so the somatosensory system in the whisker representation is very much used, and the reason is that it's a system which is very much used for plasticity, and you have a very well-defined set of centers, a very good and well-known cortical representation. So it's a very good model to study touch. I mean, it is in rats and also in mice. And so regarding with the first question, whether they carry information that is not found in spike rates, the answer to the question is yes. And this is one experiment that we did, which I think can illustrate this. So it's an experiment where whiskers sample objects, surfaces of different roughness, and do like 6 things from nothing, so free whisk, to surfaces that were very smooth, like CD, or this P1200 sandpaper, which is smooth, and then also there are some papers of different roughness, I mean, more rough. And they were recording, my colleagues here, Matthew Diamond colleagues, they were recording both from the ganglion, which is the first spiking stage after the whisker follicle, and also somatosensory cortex, which is kind of this barrel cortex, this later stage, central stage of processing. And to cut a long story short, what we found is that if you consider stimuli that have very large difference in texture, for example, here in a plot SCD, which is a very smooth surface, and this P280, which is a rough sandpaper. And if you look at the spike rate distributions, you see that typically you have more spike rates when you have rougher surfaces, and less when you have more smooth surfaces. And in general, we find that large difference in texture are represented by spike rates, spike counts. So spike counts average over the whole whisking cycle, which is about 100 milliseconds or so. But if we consider differences between, I mean, fine difference in textures, so differences between textures that are relatively close to each other. For example, if you look at the difference between this P400 and this P1200, which are two kind of smooth, relatively smooth sandpapers, then you find in many neurons something like what is plotted here. I mean, it's a sequence of a real neuron. So this is time over the whisking cycle at a given moment. And in each row, you see a different spike train in a different whisking repetition. And across different trials and different textures, different surfaces, spike counts are more or less the same. In almost all trials, you get 3 spikes in this window. But if you look at the distribution of spike rates, you have that with a very high reliability, essentially almost perfect. You get that spike patterns to one type of sandpaper elicit like an earlier response, and spike timing with another surface elicit, like, for example, a different kind of pattern of interspike intervals. And in general, I mean, this is a general result. We try to quantify it over different regions, ganglion and cortex, over different surfaces using mutual information. Mutual information quantifies in bits how well you can reconstruct which surface it was presented based on the presentation neural responses. And if you do discriminations like these ones between, for example, smooth object and rough, so between one smooth object and one rough object, so this is pairwise mutual information, so the set of stimuli is made of only 2 stimuli, you find that you have a lot of information in spike counts. I mean, essentially almost perfect in the ganglion, and I mean, not perfect, but good in cortex. But if you look instead of pairs of some paper that were not very far from each other, so some paper with similar kind of roughness, you don't get much information in spike counts, but you get a lot of additional information in spike timing measured with a few millisecond precision. So essentially what you find is that spike counts encode something like coarse stuff, and spike times encode fine stuff, fine texture differences. And this is also something that we found across different modalities. There seems to be a kind of multiplexing. So neural responses register at a fine precision usually carry some kind of information, and neural responses at the coarser precision carry a different kind of information. So it's not that all the spike times are important, or all the spike times are important, but they seem to be both important from the encoding point of view. This is a kind of what we call multiplexing. So each part of the neural signal, each time scale carries somehow different information. So, but these results here were results where there is a pattern of whisking, a dynamic pattern of whisking, because it's essentially a whisker movement back and forward. And so there is essentially a dynamics of response. So one possibility is that this spike timing somehow represent just a temporal variable, that is the dynamic of whisker movement, and the whisker deflection says the sensor, so the whisker makes contact during active touch with the surface. And this, I think, it's I think a very likely possibility, which is fine, because I think you would expect that spike timing somehow carries information about the timing of activation, and you also expect that different kind of surfaces are translated during active movement into different patterns of activation. But another thing that we try to address is whether you have also transformation between spatial variables, so definitely non-temporal variables, into spike timing, which is a kind of a different matter, and I don't think there is any expectation or need that the brain would be able to do that. So to study that, we looked at the coding of whisker location. Here, the whisker were not moved naturally. They were deflected with a very sudden, very fast ramp movement. So from rest to a certain position, one at a time. And we computed information about the location of the stimulated whisker. So all whiskers were stimulated with the same sudden dynamics, at time 0, very sudden deflation. And we look at the location, I mean, the coding of the location of the simulated whisker. And to cut a very long story short, we computed mutual information between which special position was stimulated and the neural response. And the higher it is, the more information you can get from neural responses about the stimulus location. And if we sample spike timing with a precision of 2.5 milliseconds, we found that on average across neurons and sessions and so on, spike timing carry about 50% more information than spike count. So spike timing had something that was not available in the spike counts about stimulus location. But then if we look more closely at the spike timing information, we found that essentially all information in timing was contained in the first spike timing. So if we remove all spikes from the spike train, but just apart from the first one, and we just gained information from the latency of this first spike, essentially we didn't lose any information. And for some neuron, like this very nice example neuron that we reported in the papers, of course not all neurons are as nice. Essentially, we found that in many cases, the neuron would emit just one spike per deflection, but what would distinguish one whisker from another was, to a large extent, the latency of the whisker being shorter, for example, for the principal whisker, and just a couple milliseconds longer, 3 milliseconds longer for the other whiskers. So essentially, to cut a long story short, a summary of this stuff is that there seems to be information in the very precise timing of neural activity, information about texture or also other type of somatosensory information, and that there is a kind of multiplexing, so there is some information at different scale that you don't find in another scale. And if you look at timing information, you can have also a conversion between a spatial variable and the temporal variable, and it's represented in a form of latency code. I mean, that one. So one spike, very informative, and its timing with respect to touch, essentially to stimulus variation and so on, seems to carry a lot of information. And this, as I said, has brought, when we published this result, a lot of questions about whether this format is really usable by other neural systems. And there are two issues. One is that the animals there did not, we didn't have any perceptual report about whether or not this stimulus was perceived correctly and what was the relationship to that perception and spike timing. So the behavioral relevance of this was not clear. And also, even from the conceptual point of view, a good question is how is it possible to decode this stuff if we cannot measure the latency of the neural response? It's very easy for other experimenters because we know when we stimulate the whisker or when the movement initiates, but the brain might not know about it. So to demonstrate that, we made very simple models. And my computer is frozen, so I try to, okay. And just to prove that you can decode the spike timing latency if, I mean, without needing of an external clock. And to do that, we used this columnar organization of barrel cortex, where essentially you have neurons that are clustered together very close, and this kind of barrels. Each barrel represents one whisker. And what we did, we hypothesized that the identity of the stimulated whisker can be detected when you have a small number of spatially closed neurons, or neurons in this barrel, that are activated within a millisecond or 2, 3, 4 milliseconds, whatever. I mean, this is the parameter. So essentially, if you do that, you would have a code which would require very fine latencies, and that could potentially reveal the identity of the simulated whisker without needing of a clock. And to give an example, here we plotted data from about 100 neurons in each of 2 barrels. Sorry, this is neurons in 1 barrel, but with 2 different simulated whiskers. And what you see is that if you put a very fine window of a couple of milliseconds or so through the data, and this is plotted systematically, but you can see it here in a single trial, you have a lot of spikes being emitted together by the population within a very short window, only if you are very close shortly after the stimulation of the whisker principal to the barrel. If you have spontaneous activity, or if you stimulate another whisker, then you don't have this very sudden co-activation of many neurons together. So in our view, this would be a way to read out a latency code. And to prove that this was right, we try to decode information, either doing this kind of calculation with a very, very precise, like 2 milliseconds or something, temporal precision, so really a code that detects the latencies of the neurons, or instead using like a very large window. And if you use a very large window, you could think that you could average away the noise of spike times if you think that they're noisy. If you use a short window, you could think that instead that this stuff, this very precise timing carry information, and you can use it without needing a clock. So if you use this kind of stuff to decode the activity, not only which whisker was stimulated, but also exactly when, we found that if you use this very short latency coincident detector, we could do it across trials. So each dot is a trial, 100% correct with a precision of less than half a millisecond. So these are this data. If we did it with spike rates, we could do it with instead a much smaller precision of, I don't know, 50, 60 or something milliseconds like this. And also this result was invariant to knowing when the stimulus was applied. So if you do it even with a large error in when the stimulus was applied, you still find essentially a perfect discrimination result. So this would kind of prove that this essentially very fine millisecond scale latency code can in principle be used for behavior because it can be read out by another downstream neural system. But then you need to kind of prove or show that it has a measurable impact on behavior. And to do that, we did first some kind of statistical analysis and then some kind of perturbation analysis. So we show them in order. And the statistical analysis was based on a concept that I developed in my spare time. And I think it's a trivariate extension of information theory that tries to understand not only if you encode information so if neural activity encodes information about the sensory variable, but also whether this is used for choice. So essentially we use these partial information decompositions, which is a technique introduced a few years ago by mathematicians, and that allows essentially to break down information into components. And what we did was to find essentially isolate the part of stimulus information that is relevant to choice, or equivalently in mathematical terms, the part of choice information that is related to the stimulus. And if you can isolate mathematically that component that our algorithm can do, you can reconstruct how much of sensory information neural activity informs choice, or equivalently, how much of choice information is due to the stimulus signal encoded by the neurons. I think expressing it this way is the simplest way to conceptualize this in terms of sensory information. So if you have a significant amount or a high amount of this intersection information, you can conclude that your neural code, so the information, the stimulus information that you see in neural activity, has a measurable impact on the behavioral report. So once we did this mathematics that was presented as a talk at NIPS a few years ago, we tried to apply it to empirical data. And the empirical data was a task where we did not only have a stimulation with textures similar to the one that I presented before, but also a behavioral report of the animal. So the animal essentially was on a platform. This is again an experiment done by Matthew Diamond, and had to touch a surface, and you could only touch it with the whisker. There was no visual cue, and there was also no other cue that the rat could use. And after sweeping its whiskers on the surface, the rat had to touch left or right depending on whether the texture was of one kind or another. So by monitoring the choice of the animal, we could somehow know whether the stimulus was perceived correctly or not. And also we had a high-speed camera on the top, and we used that to define to control its touch, define touch onset, a touch offset, and then we look at activity trigger to touch onset, a bit like in the previous experiments, but now activity 0 is when the stimulus touches, when the whisker touches the surface to be discriminated. And the recordings were made here in primary and somatosensory, in primary and secondary somatosensory cortices, whisker representations. The results were very similar between primary and secondary, and I think here I present only primary. And so if you look here at an example neuron, which I think is quite representative, what you see, the first thing is that, it's a bit difficult to see here because we collected many trials together and we removed the trials with 0 spikes. But there is, in most cases, just one, maximum 2, but most times 1 spikes per touch. So you have these spikes at variable times, but there is essentially 1 spike per touch. So this is again essentially, if you look at timing here, it's a latency code, because here 1 spike timing means latency with respect to touch time. And if you look at like here the trial average response probabilities, which will be easier just to form an intuition, you see that there are pairs of stimuli for a given neuron that are discriminated maybe by the timing, because here, for example, you have the same integral between the red and the blue stimulus. So same spike rate over the touch, which lasts about 50 milliseconds, but different timing, essentially 7 milliseconds latency from touch for one surface, and I don't know, 15 milliseconds here for another one. But there are also other stimuli that are differentiated by the rate, for example, this green one, because it has overall a lesser spike rate than others. And to kind of do all these analysis and see whether timing is more important than rate, first we did a calculation of spike timing and spike rate information, which is a little bit different than before. So we had essentially a rate template, which is was a flat function. So essentially here we weighted the spikes the same way regardless of whether or when they were emitted, or else we had the timing template that weighted the spike differentially with a plus or a minus with this weight depending on where they were emitted. And this timing template was done to optimize essentially sensory discrimination, so discrimination of the surfaces. And importantly, we did this timing template with a zero average, so that essentially this timing template had none of the rate information. So this was essentially, this timing template captured the additional information that you have above and beyond the total number of spikes. And if we did this kind of calculation, we had that sensor information was essentially the same between spike rate and spike timing. So they both added some information. I mean, this is a small number of information per neuron, but firing rates are very low, and this is all significant. So on average, the neuron carried information. But if you look at timing information, impact on behavior, so how much of this stimulus information goes into behavioral choice, and those numbers are directly comparable because they're both in bits, we find that essentially almost all stimulus information encoded in timing made it to choice, so was perceived, but only a very small fraction of rate information made it to choice. So for this particular task, I would say we were successful improving in this natural texture discrimination task. We were successful improving that timing is useful for discrimination, and I think spike rates seem to be instead less important. So I would say timing definitely at the millisecond level. This is millisecond precision timing, because if we scramble the spike just by a couple of milliseconds, we would lose both stimulus information and this behavioral choice information, intersection information. So timing was able to determine perception. It definitely seems to be used by the system. And to cut a long story short, I mean, this is a statistical analysis, so I think it's nice because it relates to the animal choices. But at the same time, you might say that this stuff is epiphenomenal because you don't have a causal proof. And one way to do a causal proof is to use optogenetic to manipulate the properties of the neurons. And here I think we made a kind of a slightly indirect manipulation, because my collaborator Tommaso Fellin discovered that if you had, if we regulated, if we manipulated either activating, so with blue laser, or inactivating with yellow laser, layer 5, sorry, there is a mistake here, pyramidal neurons, then it would be able to regulate the temporal precision of whisker coding of essentially all the other neurons, pyramidal neurons in different layers. And so essentially, if you had activations, this is essentially spike clusters, and unperturbed, when you stimulate the whisker, you have this kind of columnar, very precise responses that I talked about. And if you put a laser to excite these layer 5 neurons, they cut out essentially the spikes that are not stimulus related, so they enhance the precision of the response. If you instead inhibit those neurons, you lose this temporal precision, and so you are not able to decode neural activity anymore. I mean stimulus application, whisker stimulus application neural activity anymore. And this is a manipulation of the precision of the neurons, although not direct, but indirect, because you manipulate this precision through other neurons. And if you do that, you do that while you ask a mouse to discriminate whether or not its whisker encountered a pole during active exploration of the environment. What we found was that so they record the neural activity as the mouse was doing this task, and if you did that only essentially with pulse or just touch, you had this kind of precision. If you do it with laser, so inactivating those neurons, you get a reduction of the temporal precision of the neurons. And importantly, if you do that, you find that when you decrease this temporal precision through this manipulation, you also increase the behavioral reaction time of the mouse in reporting whether or not it sampled the I mean, through leaking, whether or not it sampled the pole. So there seems to be a behavioral, essentially, there seems to be a statistical correlation between the fluctuation of information at millisecond scale in spike timing and neural activity. And there seems to be also some kind of causal evidence that these manipulations affect the confidence with which the animal is able to report. What it has been asked to perceive in somatosensory terms. So, to summarize our work in somatosensation, I would say that over the years with colleagues like Matthew Diamond and Tommaso Fellin, we found that in the somatosensory pathway, both spike timing and spike rate carry somatosensory information. And spike timing carry information at a very high temporal precision between 1 and say maximum 5 milliseconds. That complement that of spike rates. And the spike rates, in many cases, carry information in a latency format, which counterintuitively can, however, be read out without external clocks or pure internal clocks, even better than spike rates can do. And also the spike timing seems to make a larger contribution, the spike rates, to behavioral discrimination of complex texture. And we also have some causal manipulation that at least is compatible with this hypothesis. And as I said, I worked a lot on other sensory modalities doing similar work. And I would say that, if we have to kind of summarize things that we found that are similar and different, I would say that one difference is that there seems to be definitely, both from perturbation experiment and observation with different stimuli, a difference in the precision of timing across modalities. Somatosensation is by, some distance the best, in that it's the one that requires higher precision, a few milliseconds really, then hearing comes second. Essentially 10 millisecond stuff. Olfaction, this is cortical data, so olfaction is a little slower, and vision is also even a little bit more lower. But all these modalities carry information. And importantly, in all these modalities, we found evidence of multiplexing, that you have some sensory information at some scale and other information at other, maybe slower scales. And then another thing that we found is that that's true for sensation, but you can also look at decision and association areas involved in generating perceptual reports or perceptual decision making. And one thing that seems to happen, not only from our group, but also from other groups, is that we found that in decision association areas, you have the generation of long time scales that are probably better to generate a decision that involves a motor output, because motor output happen on a slower scale. And so that's what we found. And another thing that I presented is that I presented the case where we had similar sensory information, but different readout, and in somatosensation. And we investigated this further in other modalities, because I wanted to see where we could challenge the idea that it's just better to have more sensory information. I mean, it's an idea of optimal coding that goes back to Horace Barlow and so on, which has a lot of influence on neuroscience. And what we found, at least, that in association areas, not in sensory areas, there seems to be a dissociation of what is optimal for encoding and what is optimal for readout. For example, having temporal correlation, which increases temporal redundancy, seems to be bad for encoding because you reduce what you can encode over time, but this seems to help the readout of this information. And this, in our view, goes back to the theories of Emilio Salinas, Terry Sejnowski, and others in the past that has convincingly shown that if you have a temporal structure in an input to a neuron, you are more likely to make sure that this information in the dendritic tree propagates and maybe then can also have a higher impact on choice. Definitely, we found in a recent paper in Nature Neuroscience that this redundancy in the neural code seems to increase at the behavioral level the efficacy of the readout. And so also something that we seem to conclude, I mean, in these recent studies, that the design of the neural code is probably, I mean, it's nice to look at information about sensory human neural activity, but the design of neural code for perception is not only based on optimizing information encoded in neural activity, so information representation, but maybe it's design of somehow leading to a good trade-off between the richness of information encoding and its safe transmission through to the behavioral output. And I mean, one theory that I have without much proof is that this balance, this trade-off may vary between sensory and association areas. For example, sensory areas, you maybe carry, I mean, you worry less about behavioral output, but if you have an association area, you have less overall information to encode because you only need to encode information which is relevant for behavior, for the particular task at hand, but you really want to make sure that this information makes it to the output. So, okay, I think this was it. So I would like to thank you guys for listening. I don't know if I went over time, I hope not. And if you have any questions, I'm happy to take them.
Yeah, I have a question. Stefano, it was a beautiful talk and the results are really spectacular. I question this number 20 to 100 milliseconds for vision because back this is like at least 10, 15 years ago.
Yeah.
And work that was done in, this is recordings from area MT, the motion area.
Yes, you're right.
In Bill Newsom's lab, when it was analyzed.
Yes.
If you look at the spikes, it looked just like the Bain and Sejnowski results with very precise timing. So I think that there may be some parts of the visual system, at least, where millisecond level of spikes is important. It's just that maybe people are looking somewhere else, in the visual stream.
You are totally right. And I think the thing that I didn't say here, I apologize, but I prepared the talk quickly and I didn't realize that I didn't say that, is that this is, I mean, first, maybe there are newer results after those of Jonathan Victor that maybe should be updated, but one key issue is that here I was reporting about encoding of static variables. So if you have an image, flash, try to reconstruct which image was shown and so on, then this seems to happen on lower time scales. But if you have a time-varying stimulus and you need to encode also the temporal structure, the visual system and all the other systems need to be incredibly quick, even, I mean, in non-primary sensory area. And your paper with Tom Albright is an example of this, I think. I don't know if it was, if you weren't, I don't remember, but that paper, I think that you refer to, I think it's an example because this was MT, so a secondary visual area, and these changes in motion direction were tracked with 2, 3, 5 millisecond precision.
Right. No, you're right about that. You have a good memory, actually. That was that was a fluctuating Gabor. So actually, I did have a comment about the whisker. So the whisker is a very interesting mechanical device in the sense that it's stiff, and it's if you have a rough surface, it sticks and it slips.
Yeah.
And I think that the mechanical properties of the whisker may actually be responsible for this an incredible precision that you've observed.
I think so. I mean, I agree with you, and I think there are people that work on this. I mean, it's not my specialty, but I think it's true. But maybe also would like to think also in general terms, and I wonder why you really need with whiskers or tactile stuff to have, especially with whiskers, such high precision, even for non-temporal variables. And maybe, I mean, one reason to have short latencies is that any stuff that you touch is closer to the body than stuff in vision. And also you have a limited number of sensors. I mean, with vision, you have so many retinal cells. I mean, with whisker, you have a limited number of follicles. And so you need probably to exploit a lot more the relative timing and temporal differences and so on. I don't know if it is a good question, but I mean, definitely there is some good mechanics in the whiskers and the follicles and so on that supports this precision. I think if people record from this ganglion, they go to like 0.2, 0.3 millisecond precision of spike time. So the precision of encoding of information is shorter than the duration of the spike. But I just, you know, the way I thought about that is that if you have a system with when very close to the body and with a limited number of sensors, then I think maybe you are kind of forced to use timing and especially relative timing to encode more information, sufficient information, and everything has to happen fast. Also, the latency here, I mean, in cortex, it's a latency of 5 milliseconds. Active touch in an awake animal. So, I mean, I don't know, in LGN, I mean, V1, I think latencies are around 40, 50, 60 milliseconds, in my recollection, even with strong contrast. So I guess there is probably just a need to process information in a different way.
Right, but by the way, there is a literature on human somatosensory timing. Ken Johnson used a stimulus, you know, with that, like for Braille, where you go across the tip of your finger, and he found, again, millisecond timing in the cortex S1 in monkeys.
Okay.
Ken Johnson, that was done like 20, 30 years ago, probably.
Okay.
And also, if I may follow up here on Terry's question. So, by the way, great work, Stefano, it's wonderful work. So in the hearing domain, so it seems that, I mean, 5 to 20 milliseconds, that's a gigantic time on the time frequency scale that for audition, right? And in fact, in some animals, because barn owl, for instance, for auditory localization, the resolution of resolutions in the microsecond range should be somewhere right in the cortical representations. So it seems that 5 to 20 milliseconds is an order, several orders of magnitude up there, right?
Okay, thank you for the remark. I mean, okay. Also, I might say this is a summary of my experience on this. So I read with our work. I mean, so it does not mean that my experience is, is representative. I mean, it's limited, of course. So, okay.
So you have to be careful because Gert, what you're referring to is the sub-millisecond resolution subcortical within the, I think it's the superior olive, which is capable of taking time differences and converting it into location, right? Auditory location. Okay. But I think it's when you get to the cortex, I think these numbers here represent cortex. Is that right?
Yes.
Yeah. Okay. So it's a little bit different when you get to the cortex, and I think that's what they're referring to here.
Yes, but so cortex can't quite get at microsecond resolution unless there is something in a spike time somewhere. Some spike times need to resolve well, not really. The zip resolution needs to be somewhere in the representation in a temporal representation, right? It's
there. It's a question of how it's used, and I think the beautiful results we've heard is that and you can really make a strong case in the somatosensory system now that those relative spike timing, carries a lot of information that is actually used to make choices and change behavior. That's what we've needed all along, right? Because everybody talks. Information's there, but is the animal actually using it?
Okay, and I mean, one thing I wanted to add. Tony Zador did some very nice experiments, putting pairs of electrodes in somatosensory or
Auditory.
auditory or visual cortex, and they trained, I think, rats, if I remember correctly, to discriminate the shortest latency between the activation, the electrical activation of those two locations. So they had to report whether it was simultaneous activation or with a delta t. And if you put the electron in somatosensory cortex, the minimal delta t is like 2 milliseconds. They can discriminate. And in auditory cortex, it finds like 10 milliseconds. And in vision, a little longer, like 20, 30 milliseconds. I don't remember the precise number, but I was impressed that what he found, no sensory stimulus, so just essentially ability of neural circuit to detect interspike intervals. That seems to match what I present here, but in terms of ranking the areas and also in terms of the actual numbers in terms of milliseconds. And we did a similar experiment in olfactory bulb, not olfactory cortex, with optogenetics, so it's different. But again, mice seems to be able to discriminate activation, essentially difference in timing activations of the order of, I don't know, 20 milliseconds, stuff like that.
Okay, Stefano, we have a last question from the chat. It is from David Murphy, who asked, there are oscillations in the brain that might imply that processing becomes synchronized, even if milliseconds timing is required close to sensory input or motor output. Do you have any thoughts on how to reconcile these two regimes?
I'm not sure I understood the question because the audio was not so good in my ears. But I think if the question is whether there is somehow a relationship between this precise timing, these packed times, and oscillation, I would say the answer is yes. And for example, many people believe that gamma oscillation that happen essentially over 60 to 100 Hertz, something like that, they pack spikes in time very tightly, such that this idea of Salinas and Sejnowski and Christoph Koch and so on, that if you pack spikes, they make it through synapses, they have that. So I think that there is a relationship. And then also one question is whether spikes are like locked to the external events, like the stimuli as I computed, or to the internal events, for example, the phase of those oscillations. And we found evidence for both. Yes, I don't know. So I think, there is a relationship. And also oscillations, ongoing oscillations seems to entrain to the timing of the stimuli. So I think there are both of those things at play. And I think, okay.
Okay, thanks a lot. So I think just a comment, I don't have questions, but I think it's really important, the work that Stefano is doing, also to help us guiding how to design the artificial systems. And that's one of the reasons why I thought it was a very interesting talk for the Telluride audience. So
I'd like to know,
I don't know if
Chiara, I'd like to know whether any of the audience have read Dana's paper. I don't know if Dana is still there, but Dana's paper seems directly relevant to this. I don't know, Stefano, if you're familiar with it, or Friedemann. The paper that I shared on the channel, it's exactly this paper right here, which is published in Terry's Neural Computation, right?
Okay.
It's a spike code that's relative to the gamma frequency. I think gamma is about 80 Hertz, right? Or 60 Hertz, something like that.
60 to 100, yeah, depends on
60 to 100 Hertz. So this thing, what it
40 to
posits,
100.
I haven't read the paper in detail, but it posits that basically a time multiplexing, allowing multiprocessing relative to the gamma frequency. And he works out some mechanism for that. So I'm curious whether, you guys are aware of this paper. There's a lot of papers, but
So
Dana's not with us anymore, I think, right now.
yeah, so that I'm in Slack. Which channel is it in?
It's in the questions channel.
Oh, question.
Yeah, I posted the paper there. So I'm just curious, people that are experts here in spike timing and spike coding, this is directly relevant.
Yeah.
I wish we had Dana with us right now, so you could give him your opinion, your honest opinion.
I haven't looked at it, but I think it might be related actually to why is olfaction so fast, right? What Stefano showed. So I'm always wondering, because I kind of think olfaction should be slow, but if you actually use a phase code, then being fast makes a lot of sense.
Wasn't it John Hopfield himself who studied these traveling waves along across olfactory bulb and showed? I mean, there's been tons of work on oscillations and olfactory bulb, right?
Yeah, Gilles Laurent in crickets, and
yeah,
there's a lot of this phase coding.
And there's a lot more evidence now that traveling waves for all these oscillations, including gamma, sleep spindles.
Again, understanding the brain is different than making silicon, right? The constraints I keep saying are different by a factor of 10 million. So the tricks may be useful, but they may be useful at a different time scale. I don't know.
Yeah, I think it's a good point. I actually have another question, if I may, for Stefano. And this is, I mean, this was beautiful talk, and you talked a lot about these trade-offs that might underlie all these coding principles. But I was wondering if you just had information, basically, could you design, according to some criteria, an optimal encoding strategy using your tools, basically, and take this to somebody who's making a sensor or somebody who's building a spiking network and tell them, look, you should be using a spike timing code, or you should be using a rate code, or the mixture 80-20 mixture between the two.
So some people did work essentially trying to derive optimal response properties from first principles of encoding. I mean, we know the example, for example, in the visual system, I mean, Bruno Olshausen or the stuff Joe Attic and so on. I mean, so my impression is that some properties, especially in early areas, primary cortical areas, or before cortex, it seems to be relatively successful, which I take as evidence that some of the neural properties are designed to maximize information encoding about the sensory variables. But what we found is that this is one thing, but if you go higher up in the hierarchy, if you look at these middle ground areas, what they call association areas, or areas that are closer to motor output, frontal areas, there seems to be also other stuff at play. For example, the stuff of redundancy seems to be at play. If you go up the hierarchy, you tend to get stronger correlation between the neurons in the space domain, and also stronger correlation over time, so longer autocorrelation time scales, even in spontaneous activity, which are probably generated by more recurrent, recurrent excitation, I guess. And what we found in our work, trying to do the same work, same task across different areas, we did this with Chris Harvey at Harvard Medical School. What we find is that as you go up the cortical hierarchy, so you get closer to decisions, you get more correlation, more redundancy, but also for every bit of information encoded in the area, you have essentially, if you do some analysis, you find that, so correlations introduce redundancy between the neurons. So consistency, every neuron is more likely to give the same message across neurons that if you didn't have correlation, if you scramble the data. And for every bit of information that you have, there seems to be a higher probability that that bit of information makes it to the corresponding behavioral choice that, according to what neurons encode, if information is distributed more evenly across neurons, which is one of the effects of correlations. And so, if you think of biophysical models like the work of Emilio Salinas, I think it's easy to understand why, because if you have a neuron or a downstream neurons that has some nonlinearity, and in particular, for example, a short dendritic integration time constant, then packing the spikes under space or time could help. So, so I believe that probably, this stuff of designing the neural code from the point of view of information maximization is good for early stuff. but maybe it's less good for association, more complex areas, because there are more, there's more than one function that the population code has to do. Not only encode information, but make it available, I don't know, transmitting, making it context dependent, whatever it is. So I think—
Stefano, are you in the Slack?
The
workshop Slack, did you join it? Yeah, there's a couple of questions for you there in the questions channel of the Slack. So if you join the Slack, you can answer them offline, I guess.
Yeah, but I don't know how to join it. I don't think I registered for the Slack. So I'm sorry for you.
I think Chiara can help you out. I can send you an invite link. Then you can look at the questions channel.
Okay, I will do. Okay, offline. Okay, and the reply. Okay, thank you.
Okay, thanks. I think we are, the time is over. Thanks again, Stefano.
Thank you.
Just a short announcement. We are going to have a talk by Hannes Saal in a few minutes in the TAC meeting. So now we leave kind of the single track meeting and we have parallel sessions dedicated to each topic area. I will in the Slack channel there's the link, the Zoom link to join the presentation by Hannes. It's going to be about how to model the spiking behavior of tactile afferents. And maybe we'll take a 5-minute break before starting the presentation there. So after these 2 very long and interesting hours, we can, oxygenate a little bit and then be ready for the talk by Hannes. So I hope to see you there.
I'm going to stop the live stream for today, though.